Hello students, we will start with second session of stem cells. So this video lecture deals with application of stem cells. In application of stem cells in your syllabus, they have mentioned about application of stem cells in cancer treatment, diabetes, heart disease, muscular dystrophy and regeneration of epidermis. So in this video lecture, I will give you the overall view of the application of stem cells in cancer treatment. So we will study one by one. The application of stem cells in the treatment of cancer. So as you all know that cancer is the most dangerous disease by causing the millions of deaths worldwide. The type of treatment depends on the cancer type and progression and the treatment. The surgery is the first option for direct removal of solid tumors located in the area. The radiotherapy can kill the tumors by damaging the cancer cell DNA, and chemotherapy with the use of very toxic drugs helps slow down or stop the tumor growth. The immunotherapy, including the use of monoclonal antibodies, checkpoint inhibitors, cancer vaccines and adaptive cell transfer now becomes an important curative for the cancer with significantly improving clinical outcomes. Okay, this is an as I told you, that is it's a tumor cell, that is cancerous stem cell therapy can the common tumors where because of the cancerous stem cells, the tumor reduction can be done with by knowing the surface markers or alter the cancerous stem cells microenvironment and induce differentiation and apoptosis. Okay, but these conventional treatment, that is what is radiotherapy or chemotherapy, so they can retain some of the cancerous stem cells. CSC means cancerous stem cells, so the tumor can reoccur. That is called tumor reoccurrence. So now I will study what are the treatments available. The insufficient and unspecific target of the traditional therapeutic approaches, as I told you, in cancer treatment often leads to therapy resistance and cancer reoccurrence. Over the past decade, accumulating discoveries about the stem cell biology have provided a new potential approaches to cure this cancer in the patients. Stem cells possess a unique biological actions including self-renewal, directional migration, differentiation, modulatory effects on other cells, so which can be utilized as a regenerative medicine, therapeutic carriers, drug targeting and generation of immune cells. As I told you in the earlier slide, the conventional treatment, they may lead to the tumor regression. Okay. The first treatment, the therapy which is available using stem cell is stem cells transplants in cancer treatment. That is usage of stem cell transplants. Okay, stem cell transplants are procedures that restore the blood forming stem cells in the people who have destroyed by very high doses of chemotherapy or radiation therapy that are caused to treat certain cancers. The blood forming stem cells are important because they grow into different types of blood cells. The main types of blood cells are the white blood cells, which are part of our immune system and help to fight infection. Red blood cells, which carry oxygen throughout our body, and the platelets, which help the blood to clot. The bone marrow, this is the bone marrow stem cells. These bone marrow stem cell has the potential to form all different types of blood cells, the white blood cells, red blood cells and platelets. So in this transplantation, stem cell transplantation, it is autologous or it may be allogenic or autologous. Autologous or allogenic. Autologous stem cell means, which means the stem cells come from the patient itself. Allogenic, which means the stem cells come from someone else. Autologous from the patient itself, allogenic from donor to the patient. The donor may be a blood relative but can also be someone who is not related, but they are matching with the matching blood group. The that is synegenic, which means the stem cells come from the identical twin. To reduce the possible side effects and improve the chances that allogenic transplant will work, the donor's blood forming stem cells must match in certain ways. This is one treatment. Stem cell transplants do not usually work against cancer directly. Instead, they help to recover the ability of a patient to produce stem cells. So after the treatment with very high doses of radiation therapy and chemotherapy. However, in multiple myeloma and some types of leukemia, the stem cell transplant work against cancer directly. This is because an effect called graft versus tumor that can occur after allogenic transplant. Graft versus tumor occurs when white blood cells from the patient body, the donor, attack any cancer cells that remain in the patient, that is the body, patient, that is the who has taken the transplant. So after high dose treatment, this effect improves the success of treatment. So this is a symbolic picture which shows the growth of the mesenchymal stem cells, which are used in the treatment of the repair of bone fractures, immune disease, and organ transplant injection. The second stem cell therapy involved is hemopoietic stem cell transplantation. Hemopoietic stem cell transplantation is a procedure widely investigated in a clinical trials in combination with chemotherapy and immunotherapy to treat the cancer, such as brain tumors, neuroblastoma, sarcoma, and breast cancer. The occurrence of this graft versus host disease when using this allogenic source of hemopoietic stem cells remains a challenge, which is often treated with immunosuppressive drugs with less effectiveness and serious side effects. So here it is the donor where the stem cells, hemopoietic stem cells are obtained, and they are grown in vitro, they are preserved, they are conditioned before the infusion to the patient. Hemopoietic stem cell transplantation, where they are with the supportive care and they are monitored for the immune reconstitution and recovery. This is the second available treatment for cancer patient, that is hemopoietic stem cell transplantation. The next one, it is mesenchymal stem cell transplantation after cancer treatment. So MSC stands for mesenchymal stem cells. The recent clinical trials have shown a promising outcomes with no related adverse effects following the co-transplantation of this mesenchymal stem cells and hemopoietic stem cells. The ongoing multi-center trial is testing the safety, tolerability, and efficacy of mesenchymal, that is angioblast-derived mesenchymal stem cell infusion in adults who have steroid resistant, that is graft versus host graft versus host disease. The mesenchymal stem cells are also found to facilitate the recovery of injured organs and could enable the body tolerance to high dose chemotherapy, that is to improve the tumor killing cells. So again, this is the picture showing the mesenchymal stem cells where they can form different types of immune cells, maybe dendrite cell, T cell, B cell, macrophages. Okay, so they are used as a co-transplantation along with the hemopoietic stem cells. The fourth available treatment is genetically modified stem cells in the treatment of cancer. The stem cells, including mesenchymal stem cells and neural stem cells, are virally transduced to enhance the expression and secretion of soluble factors, which are pro-drug converting enzymes, which act as a pro-drug converting enzymes, are tumor toxic cytokinins, chemokinins. The former system is well known for its name. It is also known as suicide gene therapy or gene-directed enzyme pro-drug therapy. Gene-directed enzyme pro-drug therapy. See here, the enzyme releasing from the viable stem cells, that is, these mesenchymal stem cells are genetically modified with the genes where they can act on the drug which is more effective on cancer cells. The enzyme releasing from the viable stem cells can convert the prodrug to active molecules that exhibit more toxic to cancer cells. For example, 5-fluorocytosine is effectively converted into tumor toxic 5-fluorouracil following the infusion of cytokinin deaminase expressing gene in the mesenchymal stem cells or neural stem cells. Likewise, iritontanine, it's also a drug, is less potent prodrug, can be metabolized into 1000-fold more toxic compound in the presence of an enzyme carboxyesterase. So here you can insert a gene that can release an enzyme which convert the prodrug into a more toxic, more effective enzyme where it converts the prodrug into more toxic to the drug will be converted into more toxic to cancer cells. It's the fourth one. Additionally, stem cells are genetically express secretable cytokinins and chemokines. The tumor toxic, that is tumor necrosis factor related apoptosis inducing ligand, that is, it is also called TRAIL, exhibit a short half-life after its systemic delivery. Interestingly, these neural stem cells used as a gene carrier to continuously release this tumor necrosis related apoptosis inducing ligand have been proven to reduce the brain tumor metastasis and improve the survival of treated mice. It is observed in the mice. This system has been employed to treat human lung adenocarcinoma in a clinical trial. The co-expression of suicide gene and cytotoxic gene of this IFNB prevents the tumor growth and prompts their apoptosis in stem cells, shows a promise to enhance treatment efficacy. This is the growth of mesenchymal stem cells where they can be act on this. They can be formed to, you can use this mesenchymal stem cells as a that is the therapeutic agent for to treat these cells. Next one is nanoparticle carrying stem cells. That means the nanoparticles have been utilized for the carriage of anti-cancer drugs for long time. The rapid excretion from the body and the lack of targeting effect and uncontrollable uptake of normal cells are the main drawbacks in the cancer treatment. It is interesting that stem cells can act as effective vehicles for carrying nanoparticles to tumor sites. In this approach, nanoparticles could be loaded into cells or conjugated to the cells on the surface of the cells. The internalization of nanoparticles via passive uptake or active endocytosis, which depends upon the size, surface characteristic and concentration of the nanoparticle as well as the incubation time, where you can use these nanoparticles as a as a target, as a molecules which target the cancer cells. Despite the rapid progress in therapeutic cell engineering, there has been no clinical trials which utilizes the stem cells as carriers for targeting nanoparticles to tumor cells. So far it is in the research. Okay, this is the stem cells where these nanoparticles are internalized. Okay, they enhance the stem cell retention and they can be track the stem cells. These nanoparticles, they are advantageous because of this nature, that is they enhance the stem cell retention and they can track the stem cells for long term monitoring, and they are also can delivery of genes to the stem cells can be done through these nanoparticles. The fifth one, the stem cells as carriers of oncolytic virus. This is one treatment where oncolytic virus exhibit a selective replication in cancer cells. These are the viruses which are inserted into the stem cells. Okay, and release a long danger signals to activate the immune system for more effective killing of tumor cells. The stem cells could be offered as a promising carriers to protect and deliver this oncolytic virus to tumor sites. For example, the human neural stem cells, when transduced with the cracked survivin, that is a virus, in combination with ionizing radiation and temozolomide, could enhance the cytotoxicity to glioma cells in vitro and increases the survival time in that is GBM bearing mice, that is glioblastoma multiforme bearing mice. Mesenchymal stem cells were found to effectively load the attenuated oncolytic virus, measles virus, and oncolytic HSV virus, which suppress the growth of hepatocellular carcinoma in case of glioblastoma multiforme bearing mice. And mesenchymal stem cells isolated from ovarian cancer patient exhibited a comparable proliferation rates and a good potential for oncolytic measles virus carries as those from the healthy donors. And however, the efficacy of stem cell carries depends on their therapeutic effects as well as the relationship between the vehicle cell and the tumor cells. Both mesenchymal cells and neural stem cells were found to support this replication, but the large number of virus could be released from this neural stem cells. So this is a picture which depicts where the stem cells which are embedded with viral particles, and the viral particles which target the cancer cells, okay, which can decrease the growth of tumors. The next therapy, stem cell derived exosomes as therapeutic carriers. So exosomes have been utilized to encapsulate various anti-cancer therapeutic materials such as that is miRNAs, proteins, and small drugs. These natural carriers possess numerous benefits over other synthetic nanoparticles, including unique biocompatibility, stability, high capacity of cargo loading, and enhanced internalization into tumor cells. In addition, they can be easily functionalized with specific proteins or ligands on their surface to enhance target effect on tumor microenvironment. The small molecules drugs could be encapsulated after priming with exogenous material. The stem cells can uptake package of these agents into exosomes and then release them to culture media by exocytosis process. The drugs including that is doxorubicin, that is gemcitabine, and cephaloplatin have been used to prime these mesenchymal stem cells. Exosomes are forced to encapsulate drugs by using this extrusion, electroporation, dialysis, or sopanin associated method. This enables to load both hydrophilic and hydrophobic drugs as well as to control the drug loading more precisely and enhance its encapsulation efficiency. Exosomes were used to suppress the tumor growth of leukemia and myeloma cell types. So here the exosome based therapies, the exosomes which are encapsulated with the drugs can be given to the patients, and the blood of this can be isolated and can study the biomarkers through this. Okay. The next one, stem cell source for the production of immune cells, where the stem cells can be used for the production of immune cells. Chimeric antigen receptor. Chimeric antigen receptor, the cells at natural killer cell, have been successfully applied for anti-cancer immunotherapy. So these clinical grade immune cells are often harvested from the own patient and activated, genetically transduced with the chimeric antigen receptor, and constructs, expands, and re-infused to the patient. Controlling the quantity and quality of these cells for immunotherapy remains challenging, especially in the patients who have experienced a heavy chemotherapy and in whom with the higher ages. In addition, in vivo anti-tumor activity of these chimeric antigen receptor immune cells are often limited due to their rapid differentiation into short-lived effector cells. The human pluripotent stem cells, including that is induced pluripotent stem cells and embryonic stem cells, could offer an unlimited source of this purpose. Interestingly, the addition of this chimeric antigen receptor and hematopoietic stem cells exhibit more benefits in treating cancer and transplant chimeric antigen receptor, and that is hematopoietic stem cells would engraft in bone marrow and continuously generate various chimeric antigen receptor expressing human cells such as T cell, natural killer cells, monocytes, neutrophils. This combination effect of these cells would result in more strong immunity to kill the tumors. So hematopoietic stem cells, they have the potency to form all other different kind of cells like erythrocytes, platelets, myoblasts, lymphoblast, okay, basophil, neutrophil, all type of blood cells. The last one, the stem cell based anti-cancer vaccines. The production of anti-cancer vaccine exhibit a highly promising due to their high immunogenicity. So anti-cancer vaccines could be raised from oncofetal peptides, or this is cancerous stem cells, embryonic stem cells, induced pluripotent stem cell based whole cells. The production of these vaccines often involves the loading of antigen into dendritic cells, which are then used to generate primary T cell response in vivo to prime the engineered T cells in vitro for adaptive cell therapy. The single use of oncofecal peptide based vaccines cannot be provided sufficient immune response towards tumors due to the tumor heterogeneity and rapid escape mechanism of tumors. So anti-cancer vaccine based on whole cell lysates may result in better outcomes. Till now, the isolation of small population of cancerous stem cells from tumor tissue remains challenging in spite of rapid progress in the identification of these cells, which hinders the use of cancerous stem cells as a vaccine source. So here schematically I have shown you the usage of, that is, cancerous stem cells where the antigen loaded to the dendritic cells, the dendrite cells can be used to generate this T cell response, which in turn these effects of this urgent with the cytokinase that is produced because of this engineered dendrite cells can be used for the treatment of cancer. That vaccine can be based on nucleic acid, synthetic peptides, recombinant proteins, viral vectors and bacterial vectors. So this is our overall summary. For the treatment of cancer cells, stem cells can be applied in nanoparticle carrying mesenchymal stem cells or neural stem cells, oncolytic virus, genetically modified mesenchymal stem cells or neural stem cells, or it may be graft versus host disease, leukocyte reconstitution, or it may be the usage of whole cell vaccine in immunizing the overall summary of what I told you in the slides. Okay, a better understanding of fundamental stem cell mechanism will improve the stem cell based regenerative medicine and anti-cancer strategies, which is imperative for more widespread clinical utilization of stem cell based therapy. And just watch this simple video, which is cell based therapies. Okay, usage of
mechanism of action of cell-based therapies. Stem cells are self-renewing undifferentiated cells that can have the potential to differentiate into any organ-specific cell depending on their origin. They can be characterized as either embryonic or somatic or adult stem cells. They differ in their potency and differentiation capacity depending on the tissue of origin. Thus far, hematopoietic stem cells, or HSCs, and mesenchymal stem cells, or MSCs, have emerged as promising cell-based therapies for severe refractory IBD cases. HSCs can be isolated from the bone marrow, umbilical cord blood, or more commonly peripheral blood of an individual, and eventually transplanted back to the same individual, autologous. These stem cells can also be received from a matched donor, allogenic, for potential cell-based therapy. HSCs are progenitors of both the myeloid lineage, monocytes, erythrocytes, macrophages, neutrophils, and dendritic cells, and the lymphoid lineage, T cells, B cells, and natural killer cells, and are capable of migrating to damaged tissue and differentiate into immunomodulatory cells in order to restore normal gut mucosal barrier function and reduce inflammation. Like HSCs, MSCs are also multipotent self-renewing cells that are capable of differentiating into several types of cells and are present in various tissues of the body. MSCs can be isolated for clinical application from bone marrow, umbilical cord blood and tissue, Wharton's jelly, or adipose tissue. MSCs derived from adipose tissue are easily extracted in large quantities with reduced morbidity and discomfort for the donor. The underlying immunomodulating mechanisms of MSCs are still unclear. However, studies have reported that MSCs exert their influence on the target cell by associating with them in a close proximity, and not solely via cell-to-cell contact signaling. Due to their low immunogenicity, MSCs have certain advantages over HSCs in regards to therapeutic transplantation. This property stems from their low expression of MHC1 cell surface proteins, as well as a lack of MHC2 and co-stimulatory proteins. As such, the need for chemotherapy in MSC transplantation is eliminated, whereas it is necessary in HSC transplantation. Another benefit is that unmatched allogeneic MSCs can be utilized in the transplantation procedure. MSCs also offer an advantage to mid-sized transplant centers as they can be mass manufactured more readily. Both HSCs and MSCs have immunomodulatory effects that can downregulate mucosal immune reactivity and promote tissue healing. Indeed, HSCs and MSCs have emerged as a promising therapeutic option for severe refractory Crohn's disease cases, such as refractory fistulizing perianal Crohn's disease. In perianal Crohn's disease, the objective is to deposit MSCs locally in fistulizing tracts to downregulate the local immune response and promote wound healing. However, future studies are necessary to fully decipher their complex cellular mechanisms. Cellular therapies will not be limited to HSCs or MSCs. Other cellular therapies, such as induced pluripotent stem cells or iPSCs, will also likely be explored in preclinical settings. Importantly, future studies should include assessments of long-term safety, the duration of response, and the cost-effectiveness and technical feasibility of such cell-based therapies.
Okay, thank you for watching. Keep watching. So next video we'll study on application of stem cells in the treatment of diabetes.